contrast to Dr. Ross, who gave a somewhat in-depth discussion of one trial, I'm going to give an overview of some of the trials that are currently available or will be available soon. So I'm going to talk about these medications or treatments, and we'll just take them one by one. But before we do that, so Dr. Ross went over that, and he had a similar slide. I have a similar, slightly different slide talking about potential causes of ALS. I mean, it really hampers us in trying to figure out how to study treatments or cures for ALS when no one's really even sure what the precise cause is. Certainly there's a role of excitatory neurotransmitters, so brain chemicals. There are brain chemicals that activate other nerves, excite them, and there are brain chemicals that quiet them down. And these are just the normal components of our cellular machinery. And so glutamate is an excitatory neurotransmitter or brain chemical. And for instance, riluzole seems to have a role in lowering glutamate levels. It also does some other things, as it turns out. Oxidative stress, so just the normal wear and tear, all the chemical reactions that occur in our body through all the cells of our body on a day-to-day basis can produce these potentially toxic byproducts that are often referred to as free radicals. And so antioxidants, you've probably all heard about vitamin C and other things that are touted as antioxidants and whether that's helpful or not. Mitochondrial damage. So mitochondria are little components of all of our cells that are responsible for producing energy. And there are data that show that mitochondria in the cells of people with ALS are damaged. Why that happens, where that comes from, whether that's sort of a downstream phenomenon that doesn't really have to do with the underlying cause is not clear. But one of the studies I'll be talking about is sort of theorizing that helping mitochondria could be a way to help ALS. And of course, as the previous talk talked quite a bit about, was inflammation, the idea that activities of the immune system could play a role, and then neurotoxicity or toxic exposures, which I'll talk about in one of these studies. And Dr. Ross already did a good job of talking about clinical trials. So quick review, we talk of phase trials. Phase 1 are very early studies, and the focus is safety, just seeing can we give a medication or substance at this dose to people and not cause them any harm. And those are often done with normal people, or they're often done with individuals who have the disease that you're ultimately going to be studying. Phase 2 trials are the first trial that starts having a control group, a placebo group. But they're small. Their goal is still just making sure there isn't some big problem of this medicine that's going to harm people, but also start looking at whether there's effectiveness or efficacy of the medication. And these phase 2 trials tend to be less than 100 people. Phase 3 trial is the big one. That's the main important thing, the so-called randomized placebo-controlled studies that use lots of patients. Based on how big or how little a change in the patient you're looking for will dictate how many patients you're going to need for that phase 3 study. And phase 4 are sort of post-marketing studies. We'd love to get to a phase 4 ALS study, because that means we found a treatment that works well, and then describing it to people, you look at it and examine it a little more to make sure there aren't problems cropping up, or seeing if there are certain groups that do better than others. And then, as Dr. Ross also went over, the types of outcomes that we look at in clinical trials. They tend to be pretty similar among the trials. So like in the Ralta study, certainly you can look at survival, you can look at the strength, and again, one of the most favorite way of looking at outcomes in our ALS trials tends to be looking at changes in decline, or the slope of the ALS functional rating scale. What this is it looks at a bunch of different aspects of the ALS disease. So looking at people's ability to walk, to use their hands to cut food or to care for themselves, swallowing and speaking, breathing. So it looks at all these features. This is way too small to see, but it breaks it up into different categories, and there's a rating from 0 to 4, 4 being normal, 0 being severely impaired. We sort of sum it all up, and then as Dr. Ross said, on average, there's about a 1-point decline in every month that happens in this scale. So unfortunately, to start on a low note is a medicine called dexpramipexole, which There's been a lot of excitement in the ALS research community based on phase 1 and particularly phase 2 data that showed that it seemed to have an effect. Unfortunately, a large study of around 800 people from sites throughout the world, and we found out just a few months ago that that was a negative study. They could not demonstrate any advantage of this medicine over the people who got the placebo. So now let's go on to studies that will be upcoming. So last year, I think I talked about rasagiline, and there was a small study that was done, and then fortuitously, the group at University of Kansas who was spearheading this got a grant from the FDA Orphan Drug Program to do a larger study of rasagiline. So why rasagiline? So this medication can have some neuroprotective. So that's just a fancy way of saying sort of help stabilize nerve cells, maybe by helping against the oxidative stress or other means. But also it has a role in mitochondria, and it can improve mitochondrial function. Interestingly, so rasagiline is a drug that's already out and available, and it's used for Parkinson's disease. So it helps improve the functioning of people with Parkinson's disease, and there may be, it's not proven, but there may be protection of those nerve cells in Parkinson's that get damaged. We already talked about studies showing that mitochondrial damage does seem to be some factor in ALS, and then, as with a lot of medications we try, they get studied in animal models. So that small percentage of people that Dr. Ross mentioned who have a genetic form, so scientists many years ago found the ability to create mice or rats that have a familial form of ALS, and every drug that gets studied usually gets looked at that. Unfortunately, there's a lot of medications and drugs and treatments that have shown some benefit in ALS and animal models of ALS, and then when they get studied in humans, they're not found to be helpful. But there was a benefit among the so-called SOD1 animal model of ALS. So this study will happen at multiple sites throughout the country, and they're looking for 80 subjects, and most will get the active treatment. So 60 will get rasagiline, and then 20 will get placebo. And then also, similar to the Neuraltis trial, that'll be augmented. The placebo group will sort of take placebo patients from other ALS trials that have been done, because again, we really see that consistent average decline in ALS slope at 1 point per month. So by adding in almost 500 other patients, you don't need as big a study group to potentially see benefit. It'll be a 1-year study. And then the main outcome will be looking at the rate of decline of the ALS FRS scale. And then other things, we always look at secondary outcomes, looking at decline in breathing, look at quality of life, look at safety, look at their survival, and then there will be also ways to look at mitochondrial function and see if it's having an impact on mitochondria. So that study will be starting later this year. I'm not certain on the date. And our Phoenix Neurologic Associates will be a site for that. So here's a study that is based on the sort of neurotoxin theories of ALS, looking at an amino acid called L-serine. And we'll be collaborating with this Institute for Ethnomedicine, which is in Wyoming, Jackson Hole, where they study drugs found in substances throughout the world and figuring out how to use that to help medication. So this guy, Paul Cox, is a so-called ethnobiologist. And he would go out to the Amazon jungle or some island, and he would meet with the medicine men and the people in these primitive societies and find out what local plants or other substances they used to treat. And then they bring those back and study them in a modern scientific way and look to see if they can find treatments. And it's borne fruit for some treatments for HIV. And in the course of doing this, they got very involved in this ALS occurring in Guam. So in the 1950s, it became realized that people on this island would have they would get a huge amount of ALS and Parkinson's and even something that looked like Alzheimer's, and some patients would get a combination of all of those, and like 100 times higher than populations in most other places in the world. And so question was obviously what the heck was doing that. And after some deal of research, they realized that it was from this toxic substance called BMAA. So it's an amino acid, and amino acids are just little molecules that get incorporated into proteins, all the proteins that construct our body. But this one, if it gets in to make up a protein, it causes a protein that's poisonous and basically can damage the body. And if it's proteins that are incorporated into the brain, then it can damage the brain. And then even interesting still is you have this BMAA, and we'll talk about how it got into these Guam natives' diet, gets into the body, finds its way into the brain, gets incorporated into proteins instead of normal amino acids that should be taking that role, causes these proteins, they have a damaging effect on the brain cells. The brain cells die, releasing this protein, and then it gets taken up by other cells nearby. So you get some cells poisoned, and you sort of get this snowballing effect. So these Chamorro natives would eat these cycad nuts, which was a local nut, and they would eat the nuts, or they'd make a paste out of it, and they'd cook with it. And the cycad nuts would also get eaten by these, we call them flying foxes. They're really fruit bats. And the fruit bats would eat the cycad nuts, and they would get a lot of this BMAA incorporated in their bodies. And for whatever reasons, they're immune to it. It doesn't hurt the flying foxes. But then the cultural thing of these natives is they would eat the flying foxes. They would make soup out of them and eat them and even eat their brains. And so they would get high levels of BMAA. So here's a picture of a fruit bat holding on to a cycad nut. And to the right of it, you see a tree with the cycad nuts. And then I showed this last year too. And then this is sort of an example of the sort of soup broth that they would eat. So it's a really fascinating story that gets even more interesting in that the source of the BMAA from the cycad nut has nothing to do with the cycad tree, but actually has to do with a bacterium or really an algae called cyanobacterium that would live in the roots of the cycad tree, produces the BMAA, which gets taken up by the cycad tree. It's found in the cycad nuts, and people and animals eat those nuts, and people would eat the animals that ate those nuts. So that's all well and good, but that's Guam. What does it have to do with here? So it hasn't been proven yet, but there's some evidence, some research that suggests that BMAA may play a role in ALS and other diseases right here in America and other parts of the world. These cyanobacteria are everywhere. They're in the desert. They form spores, and then when it rains, they can get released. But in particular, they're around lakes. So there's blooms of these in lakes, and it gets into shellfish, for instance, crayfish, and lobsters, and shrimp, and things like that. And the idea is this getting into people's systems? Obviously, everybody's exposed, so there has to be another step. There may be some people who are genetically or for other reasons predisposed to getting problems, and then if they get BMAA in their system, it can cause problems. So some small studies in Miami where they looked at autopsies of people who had died from various reasons, and they found BMAA in some brains of people with Alzheimer's disease and ALS, and they couldn't find that in people who had died of other causes. Again, just small numbers looked at, but that's what sort of fueled this excitement. It would be nice if we had a definitive proof that BMAA was a role in ALS or how big of an issue it was, but we don't. And I think last year I spoke about a pilot zinc study using zinc to sort of neutralize BMAA, and that study we're still looking to get funding for that to make a larger study happen. The Institute for Ethnomedicine has gotten funding to look at serine, L-serine. It's a naturally occurring amino acid. It plays healthy, normal roles in creating proteins throughout our body. But the key is that can compete with BMAA. It has similar features. So if you flood the system with serine, that could sort of get into the proteins. It's like the game of musical chairs where, you've got people competing for those spots. So the body's going to try to make certain proteins, and it will grab BMAA or serine. If you give lots more serine, then the odds are going to favor serine getting incorporated, which is the normal thing, as opposed to BMAA. So this is a phase 1 study, which, as I said just a few slides ago, means its focus is safety. It's going to be 20 subjects, and we're going to look at 4 doses of L-serine. L-serine has been looked at in some other neurological diseases, and we know that in those patients it can be safe and well tolerated. At the higher doses, some people might get bloating, but that usually dissipates shortly after ingesting the serine, which comes in a little powder. So the main outcome is just making sure it's safe and tolerable. Of course, while we're doing it, we'll look at changes in the ALS-FRS, although with 20 patients, it'll be hard to make any firm decisions. And will also be interesting to look at whether the serine, how well the L-serine gets into the brain by looking at it in the so-called cerebral spinal fluid, or CSF, which is this fluid that bathes the brain and the spinal cord, which we can easily access through a test called a spinal tap or lumbar puncture. So at the beginning of the study and at the end of the study, people will have a spinal tap, so they get 2 spinal taps over the 6-month study, and then there's blood and urine tests to measure their BMNA levels at the beginning and at the end. So as Dr. Ross mentioned, every study has its sort of criteria we call the inclusion criteria. So you have to meet these qualities or qualifications to get in, and then there's the exclusion criteria. If you have any of those, then you're excluded. So looking at patients for a broad age range, there's sort of classifications that we use of saying whether somebody definitely or probably has ALS. We want a certain degree, we want people to have a certain degree of involvement with their ALS. So we have it requiring the ALS-FRS be at least 25. And then you don't have to be on riluzole, you can be on it or you can't, but if you are taking it, you just want it to be stable. So if you're going to take it for at least a month, and then we would want people to stay on that during the course of the study. And some people would be on it, some people would be off of it. And that's, ever since riluzole got FDA approved, that's sort of how we've dealt with it in research studies. Some people take it, some don't, and we just sort of try to get similar groups. If you have a significant kidney problem, then too much serine could aggravate that. Again, we've talked about in other studies wanting people to have it for at least 3 years, because if somebody has a very slowly progressive form of ALS, then that's going to sort of skew the results. It'll look like the drug is helping, when maybe it's just that person was going to do well anyways. You can't take amphetamines, and you can't be on any other study medications. So what it would entail for the study is coming in every few months for just a medical examination, questions, asking you how you feel. ALS-FRS will be calculated, blood and urine tests, and then again twice during the study a spinal tap. All right, moving on to another medication, mexilotine. So Michael Weiss, a neurology ALS researcher at the University of Washington, is working with the with the Northeastern ALS Study Group to look at mexilotine. So mexilotine is a medication that's already available. It's used to treat arrhythmias, so certain heart conditions. It's what we call a sodium channel blocker. So all of the cells of our body have certain little pores or channels that let different electrolytes in and out, and that's how the electrical properties of these cells are maintained. And riluzole, Rilutech, has similar sodium channel properties. If you look at cells and you sort of starve cells of oxygen, we call that ischemia, we create ischemia, riluzole and mexilotine have similar protective effects. And then again, in at least one animal model and one small study, when you gave riluzole to one group of animals, you gave mexilotine to one group of animals and the placebo to the other, the mexilotine group lived longer. So, as you could probably see, it's kind of a thin amount of evidence for why mexilotine might be helpful, but it's a relatively safe and well-tolerated drug and is worthy of a small trial. In one study that looked at a different disease, not ALS, but it was another neurological disease where cramps can be a significant part of it, mexilotine decreased their cramps. So as at least a side benefit, mexilotine might be helpful in cramps for the ALS patients who get those. So here's an example, here's a graph from that study. So you can see about 35 mice were in each group. And you see here those lines, it's looking at how long they live. These mice don't live very long, which makes it easier to do these studies. You can see there's a very small difference. There was 7 days survival difference, although that was something that was considered statistically significant again. So we've got some data, but hardly anything robust about mexilotine. But again, it's already FDA approved, it's not expensive, we know a lot about it, a lot of people take it, it's safe in people with heart rhythm problems, it can help with the pain in some patients who have diabetic neuropathy, so it's been studied in those groups of patients. Biggest problems are gastrointestinal upset, it can cause some dizziness or incoordination or lightheadedness, some blurred vision and tremor. So the main goal is, again, it's a phase 2 study, so safety is going to be the predominant goal, but of course we'll look at progression and also look at the cramp frequency. So there will be 60 subjects recruited from 10 sites throughout the United States. It'll be a 5-month study. The sites haven't been solidified yet. They're just about there, and this study will be starting probably in the next few months. And this study involves 1 spinal tap at week 6. Otherwise, just pretty routine stuff, blood work, EKG examination, measuring the ALS-FRS. So now, ozanezumab. I've been practicing it this morning. Ozanezumab. So anything that has a mab in it means it's sort of some antibody that has been created in the lab. So there's a lot of medications now that end in mabs. So ozanezumab is interesting. So this has been created to inhibit a chemical called neurite outgrowth inhibitor, which goes by the name of NOGO-A. So what NOGO-A does normally, that's a normal chemical that our bodies make and need, is it keeps nerve cells from growing branches out. So if a nerve is damaged, it starts sending out new branches to reconnect with other nerves that it talks to. Or in the case of ALS, we're interested in nerves that talk to muscles, the motor nerves. So why would our body have this? It seems like reconnecting would be a good thing, and it is, but you can imagine that if you had some sort of injury and your nerves started growing out branches and that wasn't checked, then you get overgrowth and you get tumors and you get tangles of nerves that would be dysfunctional. So it plays an appropriate role, but here we're trying to exploit that. We see if we can get rid of NOGO, or not get rid of it, decrease its action a little bit, can we get damaged nerves to grow better, to regrow better. So in some studies where they've looked at muscle specimens from people with ALS, they found that there are increased levels of NOGO. And then, of course, in a famous animal model, so in a small study with an animal model, they found that using this ozanezumab delayed the symptom onset as well as prolonged their survival in the mice who got it. This is, I grabbed it from the internet. It's in German, but it was just a good cartoon. So in the top left, you see the normal, here's a nerve, one nerve talking to another nerve. So that long tube here, we call that the axon. And then the round dots are basically NOGO that's normally around. So in the next line over to the right, you see there's a knife. So if something damages the nerve, damages that axon, then it's going to try to regrow, and the NOGO basically will stop that. So in the last box on the bottom right, you see that those little doodads around the circles are the ozanezumab, and that's sort of binding up the NOGO and keeping it from preventing the nerve from regrowing. So NOGO-A, inhibitors of NOGO-A are being looked at in multiple sclerosis, and again, in this study they'll be looking at in ALS. It's an potentially exciting avenue of trying to help any sort of neurologic disease where nerves have been injured. So they're going to look at patients, again, pretty similar to others, basically less than 3 months or so of progression. And so a phase 2 placebo-controlled trial. So again, the main outcome will be safety, but they'll also look at effectiveness. It's sponsored by the drug company GSK, GlaxoSmithKline, and it's just starting. And they're going to have just under 300 patients as their goal. So it'll be multiple sites. Almost all the sites are in Europe, although there's a North American site, but that's in Canada. I don't know a lot about this study. I stumbled upon it on the internet, but the study is just getting underway. Again, unfortunately, most if not all of the sites will be inaccessible to people in the United States. Getting back to the inflammation or autoimmune theories of ALS. There's a study that will be starting to look at something called fingolimod, or goes by the name Gilenya. So Gilenya is a relatively recent treatment for MS. It was the first pill form of treatment for multiple sclerosis. It came out about a couple of years ago. And what it does is it keeps certain types of white blood cells in the lymph nodes. So they're stuck in the lymph nodes so they can't get out and damage the brain or damage the nervous system. That's how it works in multiple sclerosis. So the thought is, well, maybe that'll be beneficial in ALS. There are some theoretical reasons why it could also be neuroprotective. So this very busy cartoon, basically, again, the gist of it is normally white blood cells get out of the lymph nodes, get into the bloodstream, and then get into the central nervous system, the brain and the spinal cord. And if you keep these white blood cells in the lymph nodes, then they can't do mischief. So again, this is going to be a phase 2 study, which is primarily going to assess safety and tolerability. It's not yet enrolling, and these 4 sites are the ones slated to do it. It's going to be a small initial study. It's University of California in Irvine, Georgia Health Sciences, Mass General, and Methodist, which is in Houston, Texas. So now to talk about stem cells. Obviously, these get the most hype, and in the clinic, patients are frequently asking about stem cells. And so there are some studies going on in the United States for stem cells, and they're happening very slowly and cautiously, but they're occurring, and I'll talk about them in a second. But first, what are stem cells? So stem cells are cells that have the potential of replacing other cells. So when a sperm fertilizes an egg cell, and you get that fertilization, it's called an embryo, and that grows into a person. So obviously from just one cell, you get a full person, you get a baby. So different types of cells have to be made. You need to make the bone cells, the muscle cells, the blood vessel cells, the blood cells, the nerve cells, everything. You got to make a spleen, you're going to make kidneys. And so stem cells are cells that have and then once you get that person, your baby is born, then obviously you don't want cells turning into other cells that would be potentially cancer or other problems. You don't want people growing spleens on their arms or so forth. So once birth occurs, then you lose the ability to those stem cells don't exist, and you've got cell lines that can replace themselves. So your skin's always replacing, but your skin cells are making more skin cells. They're not making liver cells. If you have a heart attack, your heart can repair itself, but it's making more heart muscle cells. So the thing about stem cells is, can we get these cells to replace tissue that's been damaged? So embryonic stem cells have the most potential, and those are the ones that are frequently in the news and controversial because those are taken from frozen embryos. So you're preventing that embryo from turning into a person that has the potential to do that. But there are adult stem cells. So actually all of us have stem cells in our systems at small levels, and there are ways of pulling them out from the blood. And then the hope is that we can take those stem cells and put them into people's bodies and get them to do what we want to do. And that's always been, so that's the promise of stem cells, is that getting them to replace damaged cells. But it ain't so simple as just putting the stem cells in and say, all right, stem cells, go and fix where the problem is. So, injecting them into the blood doesn't seem to help. Injecting them just into the spinal fluid and then hoping they'll go and replace nerves that have been damaged hasn't worked out so far. I think scientists are going to need to figure out ways to coax or trick the stem cells into doing what we want them to do. I think there's another step. But then we'll talk about some studies where, well, let's take the stem cells and actually physically inject them into the spinal cord, and then maybe they'll help there. So that's what this NeuralStem study has done. So NeuralStem is a company, and they've worked with researchers at University of Michigan and at Emory University in Atlanta, Georgia. And they started out very cautiously, and they looked at 11 patients where their preparation, their special way of preparing stem cells, injecting them by a surgeon would direct them directly into the lumbar spine, into the lower spine in people who had ALS. And basically, the first goal was to say, can we do this and not make them any worse than they were before the surgery? And that worked out well. So in this very small study, it was well tolerated. Then they took another small group of patients and injected it in the cervical spine, so in the spinal cord by the neck, a little more risky. And again, with the same goal of saying, can we at least do this and not have any big complications, not make them worse? And that was successful. And so a phase 1/2 study is being planned for later this year in Mexico. And there aren't a lot of details about this. I got this from the NeuralStem website. And I also got this from the NeuralStem website. So again, this is what the researchers at NeuralStem think the advantages of stem cell treatment will be. I mean, obviously this all remains to be proved, but the hope is that these stem cells will graft. They'll get into a region where the motor neurons are damaged, and they'll take root there, and they'll rebuild nerve cells and rebuild these important connections or circuitry for getting the nerve cells and the muscle cells to work, and then maybe even protect the existing nerve cells, neurons from further damage from the disease. Again, obviously these would all be great things, and we don't know if we can do that. Those are remains to be seen. Brainstorm is an Israeli company which also is looking at stem cells in a number of neurologic diseases. Some of you might have seen this article that made its rounds around the internet of this rabbi in Israel who had gotten stem cells and he had improved. So most of the research is in Israel, although Massachusetts General Hospital is working with Brainstorm to start a very small study in Massachusetts. So it would be at Mass General Hospital in Boston and University of Massachusetts in Western Massachusetts. So an excellent resource. So you could find out what studies are available through this website called clinicaltrials.gov. And so nowadays, pretty much any study that anybody is going to do, they will register it with clinicaltrials.gov. Because if they don't register it, when they finish the study and go to publish it in a medical journal, they won't be able to get it published if it hadn't been registered. The idea being we want everybody to have open access to this information. So you can go to it, it's a search engine just for clinical trials. You can put in ALS. You can get fancier with your searches. You can put ALS Arizona, ALS Phoenix, and it will tell you what studies are going on. There's a way to click a box so that instead of seeing every study, you can see only the studies that are taking patients. Because oftentimes you'll get studies that have been done and they've already been stopped. So those would be interesting reading to see what sort of studies have been done. They may not be studies where the results have been released yet, and they're waiting for that. But if you're looking for studies that are going to be up and coming, then you can just click the box so you're just seeing those. I'll stop there and be happy to answer any questions. Yes?
Other than Guam, are there other regions of the world where ALS is predominant?
Yeah, so the question was, other than Guam, are there other regions where ALS is predominant? And the answer is yes. There's a few others, and there's the so-called Key, or I'm not pronouncing it, Key Peninsula in Japan. So there's a little region of Japan which has a very high rate of ALS, and that doesn't seem to have to do with the cycad nut. Nobody's sure of that. There are these other hotspots. I forgot to mention, getting back to the BMMA story, is that a neurologist in New Hampshire basically took his list of patients with ALS, he's an ALS specialist, and got their addresses and then put it on Google Maps. And what he saw was that the majority of the patients clustered around lakes. So there's a lot of lakes in New Hampshire. And again, that really, the idea of this BMMA is most heavily concentrated in lakes. And if you look at the people living in those areas, they have a much higher incidence of ALS than the national average. So there are some hot spots, and researchers continue to look at that because it seems like a good way to try to get a clue. The other interesting thing is in Guam, their rates of ALS are now similar to North America because the flying foxes, interestingly, are almost extinct. They ate them almost all of them up, and their rates have gone down. And so definitely, again, verifying that it is this sort of exposure. And if they also noticed that people who moved away from Guam as young kids and say moved to the United States, their rate of ALS and Parkinson's and Alzheimer's was no different from that. So, yes?
With the NP001 trials, Dr. Ross indicated the cities that were involved, I think in general, He did not specifically say Atlanta. Which city would be the closest? Do you know that?
I don't
The
know.
trials?
So, I don't know. So it's part of the Weston ALS study group who did this project, and then Dr. Ross is not still here. I mean, you should be able to go on clinicaltrials.gov, and that will tell you the studies that were involved in the initial phase study. That doesn't necessarily mean that those are going to be the same centers. But so it's interesting. So ALS is rare enough that any one center can't do a study, or to get this, you see some of these research projects are, 60, some of them hundreds of patients. So we all have to collaborate together, and over the years, there's collaborative groups, and it started off in these weird little geographic, well not weird, but these little geographic clusters. So once upon a time there was the Western ALS group, which was just the Western United States, Texas, Colorado, the West Coast, Arizona. Then there was the Northeast ALS Association, and then there was the Great Lakes ALS Association. And now, sort of, they've all merged into each other. So here in Phoenix, I'm a member of not just the Western ALS group, but the East Northeastern, which obviously we're not Northeast. So Atlanta, Emory, where most of the ALS researchers are part of the Northeastern ALS Consortium and not part of WAL, so I don't think they would be part of the neurologist's larger study. But again, when the study is ready to start enrolling, you'll be able to see it on clinicaltrials.gov. It's a good resource, although ALS's website makes it easy too to find these studies. They sort of have their own search engine, which is less cumbersome than the clinicaltrials.gov. Yes?
If you spend time in Okinawa over a period of time, would you be more susceptible?
So the question is, if you spent time in one of these regions that had the increase in incidence, I guess it depends what sort of diet you ate. Like, did you eat the flying foxes?
I hope not.
So we're not really sure how many years and years and years of exposure, but it doesn't seem that short visits there, a year, a couple of years, would give the same sort of risks that the endemic natives had.
Thank you.
Unless you ate a lot of flying foxes. Seemed kind of nasty. All right, thank you. Okay, one last question.
I was going to say, we've heard a lot about medications that are under study for the treatment of people who present with symptoms, and then I know there's also ongoing research into, what causes ALS, but is there any research going on for the early detection of ALS so that people can start on these medicines before they really, present to the doctor with some of these severe symptoms?
Yeah, good question. So the question was, are there studies looking at ways to detect ALS early? And so that maybe treatment taken at the very earliest instance would have a much more robust effect than, giving people riluzole or any of these other medicines when they've already developed weakness. I mean, in order, before someone shows up to a doctor, before they say, hey, I've got weakness, there's already been a tremendous loss of nerve cells. So just before, even if you pick up that earliest symptom, it's still, there's a lot that's percolating. So the short answer is yes. So there are, by looking at patients who have ALS, we're looking at sort of biomarkers. So the idea would be, is there a blood test or some other test that indicates it? And so there are studies looking by getting blood samples and spinal fluid samples from patients with ALS throughout the course of their disease, and that we can see certain biomarkers that are predictive of it. The problem is, who would you give the test to? I mean, ALS is so rare, and I and other ALS doctors, we see that all the time, is that, when we see a patient in the office with ALS, it's like, even if it's early, because we're clued in on it, and we've seen thousands of ALS patients. But you see somebody who starts losing their voice, and they see an allergist, or they see ear, nose, and throat, and it takes a while for it to click, because or they start having weakness in their hand, and they go, oh, that's probably carpal tunnel, or weakness in the leg, that's probably just a pinched nerve. Because if you took 100 patients with those symptoms showing up at a primary care doctor's office, almost all of them are going to have those problems. And so the tricky part is not that we don't have a test, it's just sort of, it's hard to raise the awareness because most of the time, if we just worried about ALS in every patient presenting with weakness or some sort of problem, most of the time we're going to be wrong. Fortunately, most of the time that person's going to have something else. But there are studies that are looking at some of those things in a fashion.
When was the first case of ALS discovered? Or when to the point where they actually were able to diagnose and name it?
Well, so even better. So the question is, when was the first case of ALS discovered so that they could name it? So actually in Europe, they don't call it ALS. They certainly don't call it Lou Gehrig's disease. And they used to call it Charcot's disease. So Jean-Marie Charcot was this great neurologist in the 1800s who has his name attached to a lot of things, and he described a lot of diseases. And so he described ALS. So
Did it get that name at that time?
no, it never really stuck. I mean, in Europe, they don't call it Lou Gehrig's disease, but they tend to call it motor neuron disease,
Is that what they
which ALS is a form of. So they don't have an ALS association, they have a motor neuron disease association. But sometimes it would be referred to, probably they didn't refer to it Charcot's disease because it got confusing because he had so many frickin' diseases named after him. But so in Europe, earlier times, so he described it. So it's been known about for quite some time.
Wow.
Thank you, Dr. Sachs.